Okay.
In the extreme south of the south of Brazil.
Yes, it's almost in Uruguay. Almost.
So, you know, I have to admit that I really didn't understand the variety of the geography in Brazil until the World Cup. And and then, because all the differences in elevation and climate and everything for all the different games was you know, interesting and challenging.
Yeah, we have different countries in one.
Yeah.
It's totally the opposite for the now that the UEFA is gonna happen with the World Cup of Qatar, that the people are very near. It's
I don't even know if they have enough fields. How many how many stadiums do they have?
Guys, we have to start.
Yes.
Okay.
The nice thought, but they told me we have to start. So people are getting into the room. So in Brazil, everybody is late. So so let's start. So welcome everybody. I thank you very much for having you here in this cancer symposium. I'm Alfeu Zanotto Filho. I'm professor and researcher at the Federal University of Santa Catarina. Got my PhD from Federal University of Rio Grande do Sul, a postdoc in the University of Texas under supervision of this Dr. Alexander James Bishop, and today I'm chairing this section, this cancer section at SBEFT. So today we have 5 talks, 3 from group leaders, from PIs, and 2 from PhD candidates. And for this first talk, it's a pleasure to introduce Dr. Alexander Bishop from the University of Texas. Dr. Bishop got his PhD from Oxford, United Kingdom, then completed his postdoc in Harvard, where he worked with DNA damage response, DNA repair pathways such as homologous recombination. Over the last decade or a bit more, Alex has been running the Bishop lab at the University of Texas Health in San Antonio, Texas, where he's been studying cancer cells response to chemotherapies, the effect of tumor suppressors such as BRCA1, the breast cancer protein 1, identifying defects in cancer cells that make them vulnerable to chemotherapies and targeted agents. And one interesting component of Alex's research is that he's integrating different pathways, the DNA repair with antioxidant pathways, and investigating how these different process connect and how cancer cells take advantage of this different signaling process. So, Alex, thank you very much for accepting our invitation to present, and I'm looking forward to seeing what you have to show today. I know part of it, but I want to see the whole story. So, Alex, the time is yours, and I'm here to get inspired by you.
Okay. Well, thank you very much. Thank you for the invitation to give this talk. And I know that my topic is a little bit different than a lot of the other topics or the theme of the overall conference. My interest is really in DNA damage and DNA repair. At least it has been. That was my background. Towards that, over the years, I have taken, as Alfeu was mentioning, a systems biology approach. So that means I don't look at things in a silo or as an individual molecular mechanism, but I think of the kind of the bigger picture of how things relate. And in particular, I like to apply that to thinking about disease and addressing big questions. I like to chase big questions. Don't always necessarily have the answer, but it's fun trying to understand and learn. You'll notice on the slide, the second author is Alfu, Dr. Zenitafilo. He began this journey that we started looking at, and so some of his data that he produced contributed to this talk, and I'll try to highlight some of those things. Otherwise, I would say that the majority of the work I'm going to be presenting comes from a current postdoc in my lab, Nick Pisani, and the rest of the people that are in my lab are listed that all contributed to this work. This is only one of multiple projects in my lab, but this is one that I'm excited to share with you. Okay, so BRCA1. BRCA1 is breast cancer susceptibility gene number 1. That's what it's kind of famous for. Actually, on a molecular level, what it's famous for, what people mostly consider it, is as a promoter of DNA repair, so double-strand break repair, homologous recombination in particular. So this was shown in the late 1990s and has largely dominated the field of BRCA1 biology. That said, as I mentioned, BRCA1 predisposes to breast cancer, and this month is Breast Cancer Awareness Month, so it's very pertinent to that. It's not only breast cancer, but also ovarian cancer, pancreatic cancer, and prostate cancer. So a very particular subset of cancers. And even within breast cancer, BRCA1 is really associated with triple negative breast cancer. So a very particular kind of breast cancer. Why? We don't know. And it's confusing because homologous recombination is a process, that DNA repair capability is a process that is involved in every dividing, proliferating cell. And yet, there are only very specific cancers that come up with a BRCA1 deficiency. So there's a little bit of a conundrum, a little bit of a misconnect here that nobody has been able to explain very well. And so, as I mentioned, I like to chase big questions. And so this has always been in the back of my mind. Why does BRCA1 predispose to triple negative breast cancer? All right, so that's kind of a background piece of information. So BRCA1 is important for development. So BRCA1 is a tumor suppressor. Many tumor suppressors have a normal function during development. So if you knock them out, they end up with embryonic lethality. So if you knock it out in a mouse, you end up with embryonic lethality. So BRCA1 patients are carriers of BRCA1 mutation, and they lose a second allele some point during tumorigenesis. If you specifically conditionally knock out BRCA1 during mammary gland development, you have a problem in mammary gland development. Actually, it's a little bit more complicated. If you knock out BRCA1 during mammary development, normal development, it has no impact until you undergo pregnancy. And during pregnancy is when you start to see the defect. And you can see here on the left-hand side is a normal mammary gland after going through pregnancy. You can see all of the luminal ducts and the end buds nicely starting to fill out so that they can start to produce milk for the pups. On the right-hand side, conditionally knocked out BRCA1, and you can see that the branches are not as filled out, not as well developed, and they aren't able to produce milk. It's interesting because what this denotes is a problem in differentiation, and triple negative breast cancers really are a disease of stem cells. They are trapped in a stem-like state, and they're very different from luminal A or luminal B breast cancers, which are more differentiated and developed before they form the cancer. So there's a bit of a difference between that. BRCA1 deficient patients often show a defect in the ability to breastfeed. So they don't produce enough milk, at least in their first pregnancy. And so that is something that kind of correlates very nicely with this developmental defect in a BRCA1 deficiency background. Okay, so I'm going to switch gears on you a little bit and tell you about another story that we published just about 4 years ago. And this was in a disease called Ewing sarcoma. It's a pediatric cancer. And the reason why I became very interested in it is that Ewing sarcoma are very sensitive to PARP inhibitors. Now, PARP inhibitors are used to treat BRCA1 and BRCA2 deficient breast cancers because of the homologous recombination defect. And so it was very curious to me that Ewing sarcoma has what looks like a BRCA1 deficiency phenotype, but it doesn't have BRCA1 mutations. So we started working on trying to identify what was the basis for that BRCA1 phenotype, the PARP inhibitor sensitivity. So for that, we went to go look at homologous recombination, and there are a number of ways you can measure homologous recombination. One way is to look at the foci, the proteins accumulating in the nucleus in response to damage to form this DNA repair. And so we can actually monitor RAD51, and we can see in this Ewing sarcoma cell line compared to a control cell line that you were not able to induce more RAD51 foci in response to damage. This is a classic phenotype associated with a BRCA1 defect. It's an inability to induce more RAD51 in response to damage. You can also monitor recombination directly with a reporter assay. This is a system where you have a repetitive piece of DNA, GFP. They're both broken pieces of DNA, but if you cause a break and they recombine together, they can make a functional GFP that we can monitor by FACS analysis. And when we take these cells and put in the translocation that drives Ewing sarcoma, we can see that there is a clear decrease in the amount of recombination that occurs. And so again, this is all published work. And we went on to actually understand a little bit more about why this whole effect was occurring. So I'm going to tell you a little bit about transcription now. So normally, when you have transcription of a gene, RNA polymerase loads onto the promoter. It does so in a hypophosphorylated state, an unphosphorylated state. And as it progresses along the gene, it gets phosphorylated first by CDK7 and then by CDK9. And as it goes along through the gene, eventually it has this change from serine 5 to serine 2 phosphorylations as it progresses to the end of the gene. So the protein, one of the proteins that is known to regulate CDK7 and CDK9 activity, inhibiting it, is FUS, or fused in sarcoma. FUS is an ortholog to EBOS-R1. And what we demonstrated in our Nature paper in 2018 is that EBOS-R1 acts exactly the same as FUS in inhibiting CDK7 and CDK9. As I mentioned, Ewing sarcoma has this fusion of EBOS-FLI1, and what we went on to show in our paper was that EWS-FLI1 inhibits the ability of EBOS-R1 to inhibit CDK7 and CDK9. In other words, it's an inhibitor of an inhibitor. So you end up with hyperactive or overactive CDK7 and CDK9. And what that means is that, first of all, your RNA polymerase is hyperphosphorylated in these 3 Ewing sarcoma cell lines compared to control. And if you actually measure transcription by incorporating ribouridine into the cells, so looking at how much nucleosides are you incorporating into the cells, you can see that there's very high levels of transcription occurring in our Ewing sarcoma cell line here. Okay, so if we deplete the EBOS-FLI1 translocation, we can see that phosphorylation of RNA pol 2 goes away. Okay, so we then wanted to try to understand a little bit more about what was going on, and this actually took us back to the very beginning of BRCA1 biology. So in the early 1990s, BRCA1 was discovered, and it was shown to be associated with RNA polymerase. That has been largely forgotten because of the DNA repair component, but it led us to understand what was going on in Ewing sarcoma. So Ewing sarcoma has, I told you, has no mutations in BRCA1. The protein levels are fine in BRCA1, in Ewing sarcoma. But the difference was in its location. It turns out that there was more BRCA1 associated with chromatin in Ewing sarcoma compared to our control cells. And if we deplete evocephalI1, it can now release. And the reason for that is because there's more BRCA1 that is interacting with RNA Pol2, or the phosphorylated form of RNA Pol2. So we could see that BRCA1 was effectively being sequestered or gathered up by that phosphorylated RNA Pol2. And this is in a co-immunoprecipitation assay. And it cannot interact with proteins that are involved in DNA repair, such as CTIP. And that's what is shown there. This is a relatively poor experiment, so a much better way of looking at this is asking if BRCA1 can actually go to DNA double-strand breaks. And so we went and measured that by CHIP PCR. And when we do that, we can see that BRCA1 is incapable of getting there. Okay, so we can talk a lot about this biology, and we published this, and you can I point you to that paper. But what I do want you to think about is what this means. Okay, what this means is that there is not enough BRCA1 to do everything. If it is bound to RNA Pol2, it can't perform DNA repair. Okay, so we began to ask the question, are there other kinds of interactions, other relationships of BRCA1 that cause the same effect that we may be able to take advantage of? And so for that, we began to look at another binding partner of BRCA1, which is the NRF2 transcription factor. Some of you may be aware of NRF2 as an antioxidant or a xenobiotic detoxification transcription factor. So BRCA1 seems to bind and augment it. And that was shown by multiple different groups. And in particular, the Guarini et al. Demonstrated that BRCA1 binds to the region of BRCA1 on NRF2 that BRCA1 binds to, and subsequently the region that NRF2 binds to in BRCA1. Importantly, that last part of the region that is bound by BRCA1 is also the same region that most of the DNA repair proteins bind to BRCA1. So this is just a very quick background about NRF2. This is the protein. Normally it's produced and bound by KEAP1 and immediately sent for degradation. But upon damage or some way of getting rid of KEAP1, the protein will very, very quickly accumulate, drop into the nucleus, and turn on a variety of gene expressions. So again, this is just showing that in response to damage, NRF2 will accumulate and turn on downstream genes like G-cell-C and NQ01, and that we can do co-IP showing the interaction between NRF2 and BRCA1 in response to damage, in this case, an alkylation damage called MMS, or methylmethane sulfonate. Okay, so there's not enough BRCA1 to do everything, and so we can demonstrate that if we look at levels of NRF2 activity in 3 different cell lines, 2 which are mutant in BRCA1 that you can see on the left-hand side, and 1 that is wild type, in response to high levels of damage, you can see a very clear difference in the ability of these cells to turn on NRF2 activity. If we then put BRCA1 into the mutant cells and ask how does that affect response, we can see that we can increase the response. So BRCA1 is supporting NRF2 activity in this case. If we ask how sensitive are these cells to alkylating agent, well, we can see that again that there's a nice correlation between levels of BRCA1 or mutation status of BRCA1 and their sensitivity to MMS treatment. Okay, fine. So we think that there's a good relationship between BRCA1 and NRF2, at least with regard to NRF2 activity. But what about BRCA1 and its ability to perform homologous recombination or DNA repair? So we wanted to look at that. So If we give a low level of damage, we can see the damage being induced by looking at H2AX here on this Western blot. We can also see it if we do immunofluorescence looking at 53BP1. So this is 10 micrograms per mil. It's a low level of damage. We get a nice clear induction of damage. And at this point, we're not really affecting NRF2 protein. We are inducing enough damage that's inducing homologous recombination as measured by RAD51. So that's good. Now, if we increase the amount of damage, now we're going up to 40 micrograms per mil, we can see that we started to increase the amount of NRF2 that we produce. And when we do that, we can see that we increase damage also by immunofluorescence looking at 53BP1. But then we looked at RAD51, and I should point out, first of all, that that with a high level of damage, we're starting to induce an interaction between BRCA1 and NRF2 at these high levels of damage when we're accumulating NRF2. And concomitant to that, what we're seeing is that we have a reduction in homologous recombination by RAD51. So that's quite a surprise. As you're starting to increase the interaction of BRCA1 and NRF2, increasing the amount of DNA damage, we're actually reducing the ability of these proteins to promote DNA repair. And we can also show that by looking at the interaction of BRCA1 and CTIP and these levels going down. Okay, again, this is co-IP. Better is to actually show DNA repair directly. And again, we use the same reporter assay I talked about beforehand, the DRGFP, where we induce a double-strand break and we ask how the cells, can they turn green? Can they perform DNA repair? So that's just an animation of that. And if we look at this is a kind of a busy slide, but I only want to point out a couple of points. First of all, we can activate NRF2 by getting rid of its negative regulator, KEAP1, and we can see a reduction in homologous recombination. So that's good. So that fits with our model. If we get rid of NRF2 at the same time as getting rid of KEAP1, we rescue this. In other words, it is an effect that is dependent upon NRF2. We can also play another genetic game where we can circumvent the need for BRCA1 by getting rid of another regulator of BRCA1 function. And again, we can rescue the effect. So that all makes sense. So we made a jump at this point and asked, well, what is so important about NRF2? And is there something important about NRF2 in the mammary gland? So first of all, we wanted to ask, well, is NRF2 actually even expressed in the mammary gland? Does it do anything in the mammary gland? So we started doing this by immunohistochemistry, and we measured levels of NRF2 over pregnancy. And yes, we can see that there's a nice increase over pregnancy in NRF2 levels, with the highest levels being just after birth. Okay. So again, we were trying to understand, well, what is so important about NRF2? So we depleted KEAP1, the negative regulator of NRF2, and activated NRF2 to ask, what does it turn on? What genes does it turn on? So this is a gene expression analysis, and we looked for differences that we saw. And when we did KEAP1 depletion, the gene that we saw went down the most was KEAP1. So that's good. We also saw a number of genes that are involved in regular regular function of NRF2. So that was fine. But when we looked at this, we did notice something that we hadn't seen beforehand. And that was a gene called B4GALT1. And so we were quite surprised by that. And we had to read up a little, I had to read a little bit about it. So B4GALT1 is interesting because it is a ubiquitously expressed gene that is a galactosyltransferase transferase for UDP. That's fine. But what got me excited was the second isoform, which is very specific to the mammary gland, which is important for making lactose or lactase. In other words, it makes milk. All right, so we then asked, do we see B4GALT1 in the mammary gland over pregnancy? Yes, we do, and it correlates very nicely with NRF2 levels. In fact, if you look at the correlation between B4GALT1 and NRF2 in mammary cancers, we can see a high level correlation. Many normal tissues show the same correlation, showing that there's a very tight link between the two, and at least 50% of cancers also show a tight link. So again, indicating that there is some relationship between these two biologies. All right. More importantly, we asked, well, maybe there's something about NRF2 that can rescue BRCA1 deficiency in the mammary gland. So for this, we use conditional mouse models, so Cre recombinase, where we're activating Cre in the mammary gland, and we can measure in wild type or BRCA1 conditional knockout or the double knockout of BRCA1 and KEAP1. When we do this in a normal wild type mouse, mammary gland develops fine, NRF2 develops fine, B4GALT1 is normal high, casein is a milk protein, and we can see there's plenty of milk being produced. So in normal wild type mouse, most of the pups are born alive and feed and are fine. In a BRCA1 deficient, like I showed you at the very beginning, we have a BRCA1 deficient background. We have a problem in development of the mammary gland, reduce NRF2, reduce B4GALT1, and problem in making milk. Most of the pups die. Now, if we get rid of both KEAP1 and BRCA1 at the same time, we have an interesting surprise. Now we get a more normal mammary gland development. We rescue NRF2 levels, we rescue B4GALT1, we rescue milk, and we rescue the pups. And this is a quantification of that data. Last thing I want to mention very, very quickly is how do we take advantage of this? So, you can get this knowledge, we can see this thing, so maybe there's something we can learn with this. Are there other NRF2 pathways that we can target? And so for this, we turn to the idea that NRF2 is also important for preventing ferroptosis. And there are various chemicals that we can use to induce ferroptosis. Zerastin and sulfasalazine in particular are strong inducers of ferroptosis. So the idea that we had was, could we induce ferroptosis, inhibit PARP1, and therefore target both the NRF2 activity and the DNA repair activities of BRCA1? And when we do that combination, we can see that there's synthetic lethality between the two. And these are two different, we've done this from a whole panel of different breast cancer cells, so we can see that. And this is showing the stats on that. And importantly, we can rescue this with either getting rid of the ferroptosis or inducing NRF2. These are all cell lines and growing the cells. So we also showed it with colony forming. We can also show that. And then if we take it to a preclinical model in mice, we can look in tumors and we can show again a synthetic lethal impact on tumor growth. And so now we're looking at that. All right, I'm just going to sum up because I've taken up all of my time. So first of all, I told you that there's not enough BRCA1 to do everything. I indicated to you, I hope you understood this, that there was a hierarchy of BRCA1 function between RNA Pol2, is greater than NRF2, which is greater than DNA repair. We also know that BRCA1 supports NRF2's role in mammary gland development and differentiation. There's a lot of questions that are still associated with that. And more importantly, we can take advantage of this knowledge with ferroptosis induction and PARP inhibitor induction to make a synthetic lethal in triple-negative breast cancer. So with that, I just want to finish by acknowledging the members of my lab that past and present that have contributed to this work, and of course my funding. And if you have any questions, I'd be happy to take them if we still have some time.
So Alex, great talk. Thank you very much. So as Alex said, does anyone have questions? We have a couple of minutes, and that would be good if we can discuss a little bit. I have one question, but I have more questions. But I wanna see if the audience has something. You can open your microphone or camera, or just write down in the chat. You can do in English or Portuguese. If you want to do in Portuguese, I will do the best to translate. So feel free to do the way you want. So Alex, I was thinking in a pharmacological perspective, and we can see the BRCA1 mutation. People who carry BRCA1 mutation develop cancer very early in a life. So you can see a 13-year-old woman getting a triple negative breast cancer, which is very aggressive. So I was thinking of some of the inducers of apoptosis, such as sulfasalazine, and I'm not sure about elastin if it will work in the clinic, but
Erastin is a
is
tool
that
compound. Sulfasalazine is FDA approved from 4 years old onwards.
so? It seems that there will be a good perspective of treating patients with sulfasalazine combined with olaparib. Is there any group doing this, or no one has thinking this way you were thinking?
So
when
this
you published
is
it?
right. So this is what we're aiming at doing with our preclinical models, and then if we can demonstrate efficacy, then the idea would be to move it to a clinical. And see if we can, because sulfasalazine is available. People take it as a pill for rheumatoid arthritis, and so it goes systemic, and so it's possible to take sulfasalazine, and olaparib is also through oral dose, so it's very feasible to do.
I also was thinking of because I have one of my research lanes. We are trying to use redox active compounds, and it's very hard to target the redox component because you can affect the normal cells as well. So, for example, auranofin, which is a thyroredoxin inhibitor, and do we have NRF2 inhibitors available, or they are still in the preclinical?
Yes, they're again in preclinical. So most of those are still again tool compounds. There are Ki-696 will activate NRF2. There are various other systems that can do that. We're very much trying to understand the biology of NRF2 because it seems that there are modifiers of its transcriptional activity. So depending on how you activate it and what proteins are binding to NRF2, BRCA1 included, will dictate which targets it will transcriptionally activate. So I think it's going to be important to understand how you do this. And when we looked beforehand, in fact, there was work that you did a number of years ago in the lab, where you did a bunch of different DNA damaging agents to activate NRF2. We didn't see B4GAL1, even though you also did MDA231 cells. So the way in which you activate NRF2 is going to be important for this.
Yeah, that's right. So I think we don't have questions in the chat. So thank you, Alex, again for sharing your results. These results it's a bunch of results, and and they are very interesting. And I like the way you connect different things. That's something very interesting in your papers, and I know from your research in the lab. So it was a great talk. Thank you very much.
Thank you.
So in the second talk, we will switch from the breast cancer side to the purinergic system in brain cancers. So I'm very happy to introduce a talented researcher, a woman in science, Dr. Elisandra Braganhol from the Universidade Federal de Ciências da Saúde de Porto Alegre. It's always hard to keep switching from English to Portuguese. Dr. Braganhol is a pharmacist, got her PhD from URGS in biochemistry, and has been leading a research group in brain cancer and purinergic system, with very important contributions to the field of brain tumors. So, Dr. Braganhol, thanks for accepting our invitation to present. And I'm curious to see what you have been studying in your lab at this time, and I'm sure you have a good time here. So the time is yours, your turn.
Thank you so much, Alfio, for the very nice introduction. You are one of my best friends, and it's an honor for me to participate in this seminar when you are the chair that are organizing. So I hope to to attend the expectations. I will do my best. So let me put my chronometer, because normally I talk too much. So do I have 20 minutes, isn't it? Okay. So today I'm gonna talk with you about the adenosinergic signaling as a key component of cancer-related immunosuppression. Let me organize this video here. Okay.
Liz, I think you have to put a full screen mode. At least to me, I'm seeing your slides.
Really?
Not in the full screen. I'm not sure about the others. Maybe it's mine.
It's the same here.
Okay, so let me stop share. Let me put again. Because for me it was okay.
It was like a full screen, so yeah.
Yeah, let me try again.
Yeah, there you go. No.
Go, okay?
No.
Not yet?
Not yet.
Oh my gosh. Stop share. Again. Show. Screen. No.
Are you seeing your presentation in a full screen?
Yes. For example, now for me, it's full screen. Maybe.
Vai na opção apresentação de slides. Pode manter a tela aqui, que você tem que mudar o monitor, que deve estar colocando para o monitor de apresentação. Se você está com 2 monitores ou um só?
Um só.
Pode continuar volta naquela tela que eu vou te direcionando a mudança que você faz.
Tá. Okay.
E se não conseguir, você pode me enviar também, professora, que aí eu vou passando para você.
Eu já mandei para o Fel, de repente tu...
Ali em apresentação de slide, do lado de animações, lá nos comandos lá em cima, no superior.
More. Hide video. Não, nada a ver.
Para mim, travou sua tela. Você continua no PowerPoint ou você botou para apresentar?
Não, estou no PowerPoint agora.
Do lado de, tá vendo que tem aí o arquivo, página inicial, animações, do lado tem a apresentação de slides.
Pois é, não está aparecendo essas opções para mim. De repente, Alfeu, tu apresenta para mim?
Tá, eu vou...
Ou o rapaz ali?
Vou baixar aqui. Eu não sei se ele tem ou se o Ibrahim tem.
Não,
Deixa eu
não,
aqui.
Eu só mandei para ti.
Tá, eu vou... Salva em PDF, às vezes, Elis. Vou tentando abrir aqui.
É que tem animações, né? Esse que é o problema, o negócio. Talvez ele está fazendo isso porque eu não baixei o Zoom aqui no meu computador. Ele está no... Vamos dizer, na nuvem, né? Show. Share. Mas que estranho. Esse negócio sempre funciona. Deixa eu tentar de novo aqui, peraí. Eu fiz um negócio aqui, talvez funcione. E agora, como é que tá?
Continua igual.
Continua igual.
Você quer que eu passe minha apresentação para antes? E quando você, talvez... Deixa eu ver
Uma
aqui.
outra sugestão é salvar como PDF e aí você apresentar ele como PDF.
Eu acho melhor isso.
Tá, então tá. Então, tu apresenta, daí eu faço a função aqui do PDF, então. Tá. Desculpa, gente. Tá.
Ok.
Fechar meu microfone.
So Alex, we have to switch the order of presentations because Elise is working to save in a different format, and so I'm gonna present my I want to give my talk first. Is everything all right for you?
Yes.
Okay. So now it's my turn. I thank Beth for the opportunity to talk about cancer in this Congress. I'm going to present the data we have been working over the last couple of years and data from some previous publications. So the title of this talk is Targeting Mutant P53 Protein Gain of Function in Breast Cancer, Role of Cytokines in Tumor Microenvironment. So as you may know, P53 is a transcription factor with tumor suppressor activity. It's codified by the TP53 gene and has 53 kilodaltons, and the protein has two transactivation domains and a protein-rich domain and an important DNA binding domain. That's what makes the P53 a transcription factor. In a regular situation, in an unstressed cell, P53 levels are kept very low due to MDM2 dependent ubiquitination and proteasome degradation. So when we have a damage such as ionizing radiation, tobacco smoke, or chemotherapy, we can have activation of sensors such as ATM and ATR, and we have a phosphorylation of P53 and inhibition of MDM2 proteins and MDM4. So protein P53 can accumulate and start working as a transcription factor in response to damage. As a tumor suppressor, P53 causes, when activated, it causes cell cycle arrest. Under cell cycle arrest, the cells will have time to fix the damage, and sometimes the cell can get back to the proliferation, to the cell cycle, or in other situations, the cells go to senescence, or maybe go to apoptosis, depending on the cell line and the stimuli. The other subject of this talk is breast cancer. We have different subtypes of breast cancers, and in this talk we are going to focus more in the triple negative breast cancer, which closely relate to the basal-like subtype in the molecular classification of cancers of the breast. So triple negative breast cancer is the most aggressive, and we don't have very effective therapeutic strategies. Most of the patients undergo surgery or only chemotherapy, while for other cancers we have hormonal therapies and targeted agents such as Herceptin. So we are talking about the most aggressive subtype of breast cancer in this talk. And when you go and look at the Cancer Genome Atlas, we can find that P53 mutations occur in most of the basal-like triple negative patients. Around 80% of the samples express mutant forms of P53, and which includes either missense mutations or truncating mutations. And most of these mutations occur with the DNA binding domain, so they affect the capability of p53 to bind to DNA and modulate gene transcription. So we have a loss of function in p53 in most of these cases. In addition to a loss of function, the mutant p53 also may have dominant negative effects by inhibiting the wild-type protein, by protein-protein interactions. And some mutants also have gain of function. What is gain of function? Gain of function is a mutant p53 that acquires new activities which are not present in the wild-type form of the protein. So this mutant gain of functions have been studied in different cancer types, with the most significant studies coming from breast cancer and colorectal cancers. And these gain of functions are involved in, seems to be involved in proliferation, migration, metastasis, metabolism, and various hallmarks of cancer are affected by, are modulated by mutant p53 with gain of functions. So there is a gap of knowledge in the field, because we cannot say p53 mutants, they are not the same. They are different each other, and they may or may not have gain of functions activity in cancer. So there is a very interesting perspective study showing that they say mutant p53 is not all one and the same. So we have to treat these mutants as individual proteins, and maybe with very different activities in cancer. So the biological question in our studies: does mutant p53 gain of function play a role in malignant phenotype of triple-negative breast cancers? So in this first experiment, we knocked down the mutant p53 using a siRNA and evaluated cell proliferation after 22 hours, and found using this MDA and B231 cell line, which carries a mutation in this position here, 248, and found only a marginal effect upon cell viability. We did the same experiment in another triple-negative cell line, which carries a mutation in the 157 amino acid residue, and also had the same minimal impact upon viability, and the same, and would also carry out a control with a p53 wild type cell line and the NCF7, and also found the same results with two different sequences of the siRNA. So this data suggests that the mutants are not involved in the control of cell viability. So what is this mutant involved in? So the next experiment, we carry out scratching assays and found the knockdown of the mutant in a triple-negative cell lines decreases cell migration in a scratching assay. We also used a transwell assay using Boyden chambers, where we see the cells in the top chamber and collect the invading cells in the bottom chamber, and found the triple-negative cancer cell lines are more invasive, as previously described in other papers, when compared to the luminal ones or cells with HER2 positive amplification. And in these triple-negative cell lines, when we knock down p53 using a siRNase, we decrease the invasiveness of these cells in both MDA and AGS cell lines. So we can conclude from this data that the mutant p53 has an impact upon cell invasion. The next step, we were interested in getting more information on what are the pathways that are modulated by these mutants that may affect those invasiveness, and migration phenotypes. So we did an RNA sequencing in MDA231 cells. We treated cells with an alkylated agent, MMS, which causes DNA damage. And MMS was treated in the presence of a knockdown the siRNA against the mutant P53, and the control only with the knockdown of mutant. And we have in the heat map of differentially expressed genes. In the first region, we have most of the genes that are induced by alkylation, but are not affected by the mutant. And these genes are involved in unfolded protein response and NRF2 oxidative stress response, ubiquitination, and some of them involved in DNA damage and repair. And in a previous paper, we have shown that these pathways are involved in survival to alkylating agents and cisplatin, for example. But what comes interesting is that we have a subset of genes here in the B region of the heat map and in the C region of heat map that are under control of the mutant P53. So when you knock down the mutant P53 and take these genes and evaluated to which pathways they are related, we found a number of pathways involved in cytokine production, interleukins, NF-kappa B, and macrophages. So most of these pathways are typically involved in secretome production and control of the high immune systems of inflammation. So it's very atypical to expect this type of results, this type of pathways being modulated by a mutant P53. So it's a very non-canonical pathways that is dependent upon the mutant. A more close look at the genes, we can see that some very important cytokines in the breast cancer field, such as CXCL2 and especially IL-8 and IL-6, that are widely described as involved in breast cancer malignancy, were modulated by the mutant P53. When you have a knockdown of P53, the mutant, we have a decrease in the expression of these cytokines. And we also use another gene, which is important in breast cancer, which is metalloproteinase 1, that is upregulated by MMS, but is not affected by the mutant. So we can conclude from these experiments that not all the inflammatory network is under control of by the mutant P53, and we confirmed this RNA sequencing experiment in using ELISA for validation of cytokine production in the culture medium of triple-negative cell lines. So in these two columns, we have the impact of knockdown of mutant P53 upon in the presence of damage by MMS, and the knockdown alone, how it does affect the basal level of IL-8 production, for example. And we have the same results for different cytokines and metalloproteinases, such as MMP3, CXCL2, and IL-6. All of these genes were downregulated when we knocked down the mutant. But so in keeping with the RNA sequencing data, the MMP1 was not dependent upon P53. So we have the list is bigger than shown here, but just to give an example of the impact of the mutant P53 upon secretome proteins. We also found that the same proteins are upregulated in triple-negative cell lines when compared to non-triple-negative ones. So when we take the MDA231, AGS, and MDA436, which are triple negatives, we have more production of IL-8, IL-6, CXCL2, and metalloproteinase 1. So it seems to be a triple negative related phenotype. And as you can see, we are showing that this phenotype seems to be dependent upon the presence of a mutant P53. So this in a previous work, we elucidated some of the phenotypes associated with cytokines in breast cancers. We found that these cytokines and metalloproteinase 1 are not associated with cell viability or proliferation, but they can modulate the cell invasiveness, such as the effect of MMP1 blocking the cell invasion in body chambers. We also found that the conditioned medium from triple-negative cell lines induce vessel formation in an in vitro model of vessel-like CD31 positive tubulus, and the knockdown of IO8 and the inhibitor of cyclooxygenase 2 and blockage of the IO8 receptor are all capable of inhibiting angiogenesis. So this data suggests that the secretome produced by cancer cells by triple negative may induce a new vessel formation in at least in this in vitro model. So one key point is it seems that the mutant p53 has gain of functions in triple negative cancers, but the effects we obtained in different assays was always partial. We never had a complete inhibition of transcription of production of a cytokine, even with very high levels of knockdown. So which pathways could be contributing to the mutant p53 to give malignant phenotype? So we tested a variety of pathways, and I'm here showing the effect of the MAP kinase side. And we found that the triple negative breast cancer cell lines have higher levels of MAC and ERK pathways, indicating a constitutive MAP kinase pathway in these cells. So we used two inhibitors, the sorafenib, which blocks the BRAF step of the pathway, and U125, which blocks the MAC activity. And sorafenib, we previously have shown that sorafenib inhibits MDA, MB231 tumor growth formation in xenografts and decrease angiogenesis. And in a second model of triple negative breast cancer, sorafenib also decreases the capability of cells to metastasize to different organs, and we have a prolonged survival when sorafenib is combined with cyclophosphamide and an alkylating agent used to treat breast cancer. So it seems the MAPK pathway is important to triple negative breast cancer. When you combine inhibitors of MAP kinase, of the ERK MAP kinase, with p53 knockdown, we obtained a better effect in inhibiting invasion and in inhibiting cell migration. So it seems these pathways are working together to promote these two phenotypes. When you go to the cytokines, we have a very similar effect when you do a double inhibition of p53, the mutant p53 and ERK pathway blockage, we can get the lower levels of IO8 and IO6 when compared to either inhibitor alone. So the next step, we were trying to understand how p53 and ERK pathway connect, and we observed that the ERK inhibitors don't affect the levels of p53. So the MAP kinase inhibition is not affecting the protein accumulation, and vice versa. When you use the p53 knockdown with a very effective knockdown, we didn't see any change in the phosphorylation of ERK and MAC. So this data suggests that ERK and mutant p53 are independent pathways regulating secretome production and malignance in triple negative breast cancer. So we are currently working to elucidate some of the mechanisms by which mutant p53 controls the cytokine production, and we are studying, we are using the NF-kappa B reporter gene and we found that knockdown of the mutant p53 decrease NF-kappa B activity, which is a master regulator of the cytokines you are studying here. We have the same phenotype when it blocked MAP kinases with inhibitors. So it seems that NF-kappa B is downstream on p53 and MAP kinases, and probably it's involved in the control of cytokine production by this triple negative breast cancers. So a take-home message from this talk is that mutant p53, at least the two mutations you are studying here may have got a function, a gain of function in triple negative cells. These mutants are more associated with invasiveness and secretome production than survival of these cell lines. The mutant p53 cooperates with ERK-MAP kinase pathway to promote production of cytokines and invasion and migration. And the NF-kappa B pathway seems to be downstream on mutant p53 and ERK. So this is our working model at this point. We don't know how mutant p53 interacts with NF-kappa B. That's one thing we are working on. But the consequence to the cancer is that when you have a loss of function of wild type p53 and you have a mutant, at least some mutants, they can promote release of various proteins involved that makes the secretome of breast cancers. And we know from a very strong background that these cytokines and metalloproteinases are mediating a variety of cancer hallmarks, promoting inflammation, invasion, migration, and many aspects of the cancer phenotype. So the pharmacological perspective is that restoring the normal function of p53, either by targeting the mutant or with gain of function or their downstream effectors, may offer an opportunity to find novel strategies to treat triple negative breast cancers. So I want to thank my lab cancer at UFSC. These guys are very hardworking, and I'm proud of them. And a special acknowledge to my PhD candidate Raquel Neves, who has been taking this project, and Dr. Alex Bishop for helping with very important experiments to this project. And thank you all. And remember that it's October Breast Cancer Awareness Month. Do your mammograms and help and do awareness to your relatives as well. Thank you. So, Dr. Zanotto-Filho is open for questions. Does anyone have questions, please?
There is some questions in the chat.
Let me see. Nice results. Did you investigate also the silence of p53 in vivo triple negative model? That's one thing I want to do. We are trying to set up some cell lines with p53 knockout by CRISPR. We know that p53 knockout doesn't work very well in some cell lines because it causes some type of responses. So you want to deal with it and see with knocking down the mutant and implanting and doing a xenograft, or we can modulate at least the secretome or angiogenesis, or something we can do in an immunocompromised mice. So it will be very good if you could do this, a knockdown of p53 or overexpress a mutant in a regular mice in an immunocompetent mice. So, but it's a way more difficult than working with xenograft, because p53 mutations in mouse are not the same as the hotspots we have in humans. So that's a limitation in the field. But thank you for asking this. You have a question, Alex?
Yes, always. So, very nice work. I'm curious, based on the pathways that you're looking at, and the availability of various inhibitors, how you would use this knowledge for treatment.
So that's a key point. There is a dozen, not a dozen, a hand of molecules that are supposed to be restoring p53 function, at least in vitro models and with some evidence in preclinical models. So I'm wanting to test some of these molecules. I don't know when I will have money to apply these interests in the bench, but I think that we can try to restore p53 using these chemicals, this pharmaceuticals, and but maybe if we identify something downstream on the mutant pathway, maybe you can use some targeting agents to block some key process downstream of the mutant. The difficult part of this of targeting mutant it's because it seems that every single mutant is a little different from the other. They are not the same. They may have some something that is similar, but they have some particular mechanisms that they modulate. So I don't know exactly how to do. I think that the next steps is to try to identify something downstream and use some molecules that are supposed to restore activity, and or we go to mRNA vaccines against the mutants. But I don't think it's very easy. If you have any suggestion, please send me an email.
You can use the nanotechnology tool that I developed. Just change the siRNA.
Yeah, that would be good. If you protect the RNA using a nanoparticle, maybe it can be done. There is an adenovirus that people are using in Japan. They are using adenovirus injecting directly into the tumor, and this adenovirus codifies to wild type p53, and it's clinically approved. So there is this type of therapy that is possible. We know in pharmacology we always want a small molecule inhibitor. That's the type of molecule we wanted to discover, but there is another approach that may be interesting.
Yes, I think so.
So I think we don't have questions. So thank you very much. So let's start the next talk. So I've already introduced Dr.
Here we go.
So it seems that your presentation is fine this time. So the time is yours, Elise. Go ahead.
Okay. Okay. Very
I'm
nice.
looking forward to see what you have of novelties to show. Thanks.
Okay. Thank you so much again, Phil, for the invitation. And I'm so happy that I saw some a big friend that is in the room, Andressa, and there is a teacher from URGS, Professor Teresa Dalla Costa. It's very nice that we are not together in the same place, but it's nice to be in the same virtual room. So let me put my chronometer. So today I'm going to talk with you about the adenosine signaling as a key component of cancer-related immunosuppression. You can go next. The main file of my laboratory is the research in glioblastoma. So glioblastoma is the worst and most common primary brain tumor. The treatment is very limited. It consists of surgical resection when it is possible, followed by radio and chemotherapy with temozolomide. That is just the only one chemotherapy available for the treatment of glioblastoma. Unfortunately, glioblastoma is refractory to classical immunotherapy, and it's a tumor very aggressive, characterized by high angiogenesis, proliferation, infiltration, chemoresistance rates, and also high recidive rates. So the median survival of patients after diagnosis is around 12 and 14 months. But in the clinical routine, around 50% of patients die after 3 or 4 months after the diagnosis. So the development of new therapeutical strategies are really needed to offer alternatives to these patients. There are a lot of studies published in the literature, published by our group also, that shows that the tumor microenvironment composed by immune cells, its secretory products, is fundamental. Is a key component of glioblastoma progression. Next. So here we have a image illustrating the microenvironment of the inflammatory microenvironment of glioblastoma. As you can see, the tumor, the glioblastoma is a kind of cold tumor. There is a few infiltration of lymphocytes. The main immune cells infiltrated in the microenvironment of glioblastoma are innate immune cells, for example, as microglial cells, macrophages, and neutrophils. Studies from our group and from others also have been shown that this microenvironment there are higher concentrations of ATP and adenosine in the tumor microenvironment. This ATP is well known to induce cell proliferation, and it also acts as the danger signal, and it helps to recruit immune cells to the tumor microenvironment. Adenosine is well known to induce immunosuppression, angiogenesis, and also cell proliferation. So next, please. The main extracellular source of adenosine is the activity of 2 ectoenzymes, CD39 and CD73. Together, these enzymes hydrolyze ATP and/or ADP to AMP, and this AMP is further hydrolyzed by CD73 to adenosine. So adenosine target its biological actions via P1 purinergic receptors that are located in the cell surface. Next. So our group has been shown that CD73 is overexpressed in glioblastoma cells. So we think about whether if you knock down or if you use pharmacological inhibitors to decrease the CD73 activity, we could reduce the extracellular adenosine levels, and as a consequence of the decreased levels of extracellular adenosine, we can control the proliferation, invasion, angiogenesis, chemoresistant, and also immunosuppression. So we started to work with the idea that if you decrease the extracellular adenosine, it could be it would be a very nice manner to block this variety of pro-tumor actions of this nucleoside. So next, please. So the objective of this project is it was to investigate the potential of CD73 as a target for glioblastoma treatment. So we developed 3 strategies that I'm going to show you during the seminar. The first strategy, it was a concept study. When we made the determination of CD73 role in vitro, and in vivo glioblastoma growth. Next, in the second strategy, we developed a nanotechnological formulation as a CD73 siRNA delivery system for glioblastoma therapy. And finally, in the third strategy, we evaluated the immunotherapeutic activity of CD73 siRNA delivery in a preclinical model of glioblastoma. So next, please. So here in the first strategy, in the proof of concept study, we first started working with glioblastoma cell lines. And for this end, we use U87 human glioblastoma cell lines and also C6. And in this culture model, we evaluate the effect of adenosine and also the effect of AMP, that is the substrate of CD73, on different parameters related to cancer malignity. We also apply a pharmacological inhibitor of CD73, APCP, and we also developed a very specific siRNA sequence. We combine the sequence with lipofectamine, and then we use this strategy to knock down CD73 in the cell cultures. We also use the preclinical model of glioblastoma, and for this we inject C6 glioblastoma cells in the Whistler rat brain, and we perform the treatment of these animals with APCP and also with siRNA specific for CD73 knockdown. Next, please. So here I bring some data using in vitro approach. And here, this is a figure with the general results when we evaluate the effect of adenosine, AMP, APCP, and the effect of CD73 siRNA on cell viability. Next, please. Next. Here, okay. So here, as you can see, here's in the left the experiments made on C6, and in the right the experiments made in U87. We treat the cells with different concentrations of adenosine for different times, and we can observe that adenosine has a different modulates the in vitro glioblastoma growth, and the effects on cell viability of glioblastoma cells depends on the concentration and the time of exposition. Next, please. We check the effect of the AMP, the substrate of CD73, and then we can observe in the blue line that the high concentrations of AMP decrease the cell viability of glioblastoma cells. Next. And And APCP, the inhibitor of CD73, also decrease the cell viability of glioblastoma cells. And finally, in according with this data and with the participation of CD73 on cell proliferation, then you can go. We also observe a decrease on cell viability on the cells that were treated with siRNA for CD73. Next, please. So here we observe that CD73 inhibition also reduce in vitro glioblastoma growth. In the top of the figure, you can see the effects of the knockdown of CD73 and also the effect of the pharmacological inhibitor of CD73. You can see that there is a decrease of the number of glioblastoma colonies. Next. We also check the effect of adenosine on cell migration and invasion using a Boyden chamber. And we also evaluated in parallel with this migration and invasion assay, we evaluated the expression of metalloproteinase 2 and also vimentin, that are proteins very important for cell migration and invasion. And we can observe that the treatment of glioblastoma cells with adenosine resulted in an increase of migration and invasion, and which was followed by an increase of expression of metalloproteinase 2 and vimentin. Next. The APCP target the opposite effect, showing that when CD73 is inhibited, we have a decrease in migration and invasion. Next. In according with this data, siRNA for CD73 also reduced the cell migration and invasion, and this data was followed by a decrease on metalloproteinase 2 and vimentin. And in the end, next, when we knock down the CD73 and we treat the cultures with adenosine, we as you can see, we can restore the migration and the invasion of the glioblastoma cells. And this data suggests that the inhibition of CD73 and the consequent reduction of adenosine levels in the extracellular, in the tumor microenvironment, are important mechanisms, are the key mechanism that is inducing the increase of invasion and migration. Next. We also check whether the combination of the inhibition or pharmacological inhibition or the knockdown of CD73 impacts the effect of the temozolomide, which is as I told you before, is the treatment used for patients with glioblastoma. And as you can see, when we perform the combined treatment, we can reduce the cell viability in C6 cells in the top of the panel. In the middle of the panel, you can see the effect on U87 glioblastoma cells. And in the bottom of the panel, you can see that the treatment, the inhibition of CD73 also reduce the IC50 for temozolomide, indicating that it would be a very nice strategy to inhibit CD73 as a strategy to chemosensitize glioblastoma cells to temozolomide. Next, please. Finally, we check the effect of the blockade of CD73 in vivo glioblastoma growth. And here in the top, I'm show you the experimental design of the experiment. Then we implant the C6 glioblastoma cell in Wistar rats. And then in the day 7, 14, and 21, we administrate intracerebroventricular the farmacological inhibitor of CD73 and also the siRNA for CD73. And as we can see, the pharmacological inhibition of CD73 and also the knockdown was able to decrease the glioblastoma progression. And you can see that in the box, we can observe the levels of extracellular adenosine that are present in the liquor of these animals. And then you can observe that the decrease of tumor growth is parallel to decrease of the extracellular levels. We also observe in this model an increase on leukocyte infiltration in the tumor microenvironment. So we think that this leukocyte infiltration could be helpful to induce apoptosis, to induce cell death of the tumor cells implanted growing in the brain. Next. So as a partial summary of this first part of this work, we show that the knockdown or the inhibition of the enzymatic activity of CD73 was able to increase the sensibility of glioblastoma cells to temozolomide. It decreased the malignity, it decreased the proliferation, it decreased the migration, and in vitro and also in vivo. So with this data, we can suggest that yes, CD73, it's a very nice target for to develop as a therapeutic strategy to treat patients with glioblastoma. Next. So here, the second strategy, we develop non-emulsion as a strategy to perform the CD73 siRNA intranasal delivery for glioblastoma therapy. Why we choose the nasal route? We choose the nasal route because, as you can see here, is a route that overcome the difficulties imposed by blood-brain barrier, and also it's a route that is very comfortable for the patient to administrate this therapy. To this end, we developed a cationic non-emulsion that is a kind of nanotechnological approach. We combine this cationic non-emulsion with siRNA against CD73, and so this treatment was first we made the physicochemical characterization of this formulation, and after we evaluate the anti-glioblastoma effect in cell culture and also in the preclinical model of glioblastoma. Next. So here we check different charge ratios to check the best situation of complexation of the siRNA to the nanoemulsion to the non-emulsion, and then we see that these charge ratios of +4 that is related to the cationic component of the non-emulsion, and -1 charge that is the contribution of charge of siRNA, is the best situation for the complexation of the nanocarrier with the siRNA sequence. Next. So here we analyze whether this nano complex could be uptaken by glioblastoma cells, and whether it could be effective to decrease the CD73 expression. Here we can see that the formulation was uptaken by glioblastoma cells, and here we can see that the treatment with siRNA against CD73 was able to decrease the CD73 enzyme activity. Next. Here we show that this formulation is not cytotoxic to astrocytes, which is very good, because we aim to develop a treatment that would be effective and in the same time selective against tumor cells, and with smaller collateral effects on normal cells. Next. We also check. Next, we started to check the delivery of this nanostructure in an in vivo system, and here we analyze the distribution of the siRNA CD73 complex after a single nasal administration in Wistar rats. As we can see, we can detect the nanostructure in the serum, in the liver, in the kidney, in the lung, but we are very happy to see that the formulation arrived to the brain, and we have the bigger quantity of this nanocarrier after 18 hours of administration. And because of this profile, in the animal experiments, we choose to administrate the formulation each 12 hours. Next. So here we check the effect of the therapy that we develop in a preclinical model of glioblastoma. Again, we implant the cells in the rat brain, and we made the treatment from the day 5 to the day 20, 15 days of treatment. We treat the animals twice a day, each 12 hours. And here are the control groups. And as we can see, the treatment was really effective to reduce the in vivo glioblastoma growth. Next. So here you can see that the decrease of tumor growth was followed by a parallel decrease of adenosine level in the liquor, and also a decrease of inosine level in the liquor. Next. We also check for the peripheral toxicity, and as you can see, the treatment didn't induce liver or kidney or lung toxicity. Next. And in the partial summary of the second part of this project, we can observe that the intranasal infusion of siRNA sequence against CD73 were able to arrive to central nervous system and it was very effective to reduce the in vivo and in vitro glioblastoma growth. Next. So we finally checked we explored the immunotherapeutic value of CD73 blockade in glioblastoma. We are very interested to evaluate this characteristic of the developed formulation, because glioblastoma does not respond to the classical immunotherapy. And as adenosine is a immunosuppressive molecule, we hypothesized that the decrease of extracellular adenosine levels in the tumor microenvironment would be nice to decrease the immunosuppression induced by the tumor cells. So again, we made the same protocol. We implant the tumor cells and we treat the animals twice a day during 15 days. And in the end, we remove the tumor and we analyze the tumor-associated immune cells by flow cytometry. Next. Here, first we analyze the control, the tumor of the control group. Here is the tumor of the treated animals. So first we analyze the percentage of apoptotic cells in the tumor microenvironment, and then you can observe that the treatment was effective to induce an increase of apoptotic cells. And we did not observe alteration on the cell proliferation using CAIM67 marker. Next. We also observe here, you can see in this square, we also observed that the CD73 expression was decreased in the microenvironment of the treated animals. And you can also observe in the graph in the letter B that there is in the letter B and the letter C, we can observe that a decrease on microglial in the expression of CD73 in microglial cells and also in tumor-associated macrophages. Next. We also observed that the treatment was able to decrease the quantity of microglial and macrophage cells. And in the bottom, in the letter C and letter D, this is not significant result, but you can observe a tendency of decrease of M2 microglia and M2 macrophage associated cells, which is very nice because M2 macrophage, M2 microglial cells are well known to induce glioblastoma immunosuppression. Next. Here we observe that in addition, the knockdown of CD73 also reduce the number of T regulatory cells in the tumor microenvironment, which is very nice also because the Treg lymphocytes are also well known immune cells responsible for the induction of immunosuppression in the tumor microenvironment. Next. And here is the summary of the final part of this work, where we have shown that the treatment with CD73, with the inhibition of the knockdown of CD73, induce an immune cell activation and a tumor regression. We can observe an increase on cytokines responsible for anti-tumor effects in the tumor microenvironment. The treatment also result in a decrease of CD73 and also in a decrease of in the adenosine present in the tumor microenvironment, a decrease in the cells, in tumor-associated immunosuppressive cells responsible for glioblastoma development as microglial, macrophages, and Treg lymphocytes. And finally, the treatment also induced an increase in the percentage of apoptotic cells. Next. So what is next? This product that we developed, we submit for the analysis of the Brazilian, for the European, for the Hong Kong, for the American Office of Intellectual Property. And in the last year, we got the patent, the Brazilian patent. And in the last week, we received the patent from the US PTO, that is the American patent office. The office, European office and Hong Kong office are still analyzing the request, but we are very confident that we'll got the intellectual property on this place. Next. And what you are doing now, and I'm in the same time, I'm quite anxious, but I also very happy because we via SEBRAE and via CNPq, we approved some projects that is going to be possible to perform a phase 1 clinical trial. So at this moment, we elaborate a phase 1 clinical trial project. We submit to the ethics committee, and now and in parallel, we're working with preparing the documents to request to submit this clinical trial to analysis of Anvisa to get the approbation to develop this trial in the patients here in the Clínicas Hospital here in Porto Alegre. Next. So here are the researchers involved in this project, the universities, and the SEBRAE and the agencies of government that are giving some research to develop this project. Next. This is my group. This is the Braganhão Lab, as my students call. And this is in the bottom of this slide is my email. And I would be happy if you have some questions or if you have some suggestions. Thank you so much. And sorry, because I think that I speak more than 20 minutes, but I'm so sorry.
Yeah, it's a little more.
Yeah, sorry.
So, thank you very much, Liz. It was a very interesting, but we don't have much time because we're running late. But
yes.
Before you get rich with these patents, we have time for one question.
Yes.
So does anyone have a question? If not, I have one. Just one.
People are very shy today.
Yeah, they should try. So while they think a little bit, I'll ask you, do you think, it seems that your formulation works better, the concept of the adenosinergic pathway seems to work better in vivo than in vitro, based on the results you have shown here.
Yes.
But, and you showed some results with temozolomide as an alkylating agent, which is not as effective as expected in the clinics. It just prolong a little the patient's survival. Do you think the combination, you see patients being treated with probably with high ionizing radiation and either or temozolomide? Do you think you can have different results when you combine your strategy based on CD33 if the patients are treated with chemotherapy, or how do you expect this classical treatment can affect your nanoparticle to work?
Yes, in fact, also I didn't bring this data because it would be so long, but we checked in the animals. We made an experiment. We made a complete experiment, treated the animals with just temozolomide, just siRNA for CD73, and the combined treatment, because I was pretty sure that the combined treatment would be better, would target a synergical effect, but we did not observe synergical effect, and the therapy, the knockdown of CD73 therapy was much more effective than temozolomide.
Yeah,
at least in the animals.
Yes, that's what I was thinking of, because we actually in the cancer field, you want to get rid of the classical chemotherapy, and if you can have a targeted therapy with a lessened incidence of side effects, that will be way better. So as we are running late, I have to move to the next two talks. So thank you very much, Elise. It was very exciting to see you're moving to the clinics. That's we know how challenging this is in Brazil. So congrats to you and your group and your institution for
Thank you.
getting to this point. So from now on, we have two talks by PhD candidates who will be here practicing their presentation skills. So the first one is swelling. Swelling Swelling Rocha Silva is from the Federal University of Bahia and Fiocruz, Bahia. Swelling has been working on the effects of an anti-leukemic compound upon leukemic stem cells. So thanks for accepting and for joining us, Swelling. So it's your moment, and let us learn a little more about leukemia. So there you go. It's your turn.
Hi, hello everybody. Good evening. So as Alfeu told us, my name is Suelen. I'm from Federal University of Bahia, and my PhD is about the anti-leukemic potential of EMT to eliminate leukemic stem cells from acute myeloid leukemia in an in vitro and in vivo model. The acute myeloid leukemia, or AML, is the most lethal among the leukemias. This is because patients, usually in 5 years, they show a relapse of disease, and this relapse is related to a small population of cells that have a limited potential of replication, and they are resistant to the chemotherapy. And the cells are named as leukemic stem cells, or LSCs. And this works as a reservoir for these relapses of disease. In these LSC cells, a lot of pathways are abnormally expressed to favor their survival. One of these pathways are the NF-κB, and the NF-κB is constitutively activated in these LSC cells, but not in normal progenitor and hematopoietic cells. So this could work as a strategy for kill LSC cells, but not normal cells. So the NIH from US, they made a screen for drugs that are in use now, and they find 19 drugs that are inhibitors from NF-κB, including EMT, that are my drug for studying in these tests. So our aim was evaluated the anti-leukemic potential of EMT to eliminate LSCs from AML in a translational model in vitro and in vivo using KJ1 cells. For this, first, we determining the IC50 for EMT in 18 cell lines using the Alama Blue method. And then for the mechanism of action studies, we use KJ1A. And we perform a lot of cell flow cytometry with some markers for LSCs, markers for cell viability, for apoptosis, and for oxidative stress. Also, we perform qPCR, and then we perform in vivo assay when we use NSG mice that are treated for 15 days with imetine, and then they are euthanasiated, and the bone marrow and blood are collected and analyzed by flow cytometry. So first, we show here the table of IC50, and the EMT treatment shows a promising IC15 for all the 18 cell lines that are tested. And for KJ1A that we use for our studies that are acute leukemia cell line, they are from 1.7 micromolar. Then we start this cell mechanism action studies. And the first was about the cell viability with 2 different markers. And in both markers, we can see a decrease of cell viability when treated with the EMT, and the increase of cell death in apoptosis. We also check if with markers for LSCs, if MT can kill the leukemic stem cells. So which shows here that the treatment can reduce the cells marked with CD13, CD34, CD38, and CD123. And all these markers are very specific for leukemic stem cells. So shows that EMT can kill this population of cells. And then we check if the apoptosis can participate for this mechanism. So we check the activation of caspases and the cleavage of this substrate, the PARP1. And we shows that there are one increase of both caspases and PARP when treated with EMT. And for confirm these results, we use a pharmacological inhibition with ZVAD that are inhibitor for caspases. So with the pre-treatment with the inhibitor, we can see that the cell death was prevented when compared with cells without inhibitor. So this confirms that caspases can participate from this cell death induced by EMT. Then we check if the compound can make some mitochondrial changes. So we evaluated the mitochondrial depolarization, and we check that there are one increase of this mitochondrial depolarization when treated with EMT. Also, we check if there are generation of reactive oxygen species, and we see that there are one increase of this reactive oxygen species in 1 hour after the treatment. And again, we check with the pharmacological inhibitor, now for reactive oxygen species, the NAC, and we check that when treated with the inhibitor, there are a prevention of cell death, indicated that the oxidative stress in the cell could participate almost in part of the death occasioned by the EMT. Then we evaluated the content of DNA in the cell and if the treatment can alter the cell cycle. So we check here that there are increase of DNA fragmentation in 48 and 72 hours. And this shows that the compound can fragmentate the DNA and accumulate this in sub G0, G1 in the cell cycle. After this, we also check if the compound can inhibit the NF-κB. So we shows that the expression of NF-κB P65 was reduced when treated with the compound for 24 hours. What we expected, so NIH shows that EMT also inhibit the NF-κB. And then we made the qPCR for 92 genes. These genes are oncogenes or gene suppressors of tumor. And for these 92 genes, 54 are upregulated, including the NF-κB inhibitor. And for the 5 genes that are decreased or downregulated, all are very low oncogenes, as twist1, as WNT, as BCL2. So this can open the window for studies other pathways in this leukemia with EMT treatment. And finally, we made the in vivo assay, and it shows that in the bone marrow and in the blood, mice shows a reduction of leukemic cells when compared with the treatment. So the EMT could be a good anti-leukemic compound for treatment in vitro and in vivo. So as conclusion, these results shows that EMT is a promising compound, cytotoxic compound, able to eliminate AML leukemic stem cells in vitro and in vivo, as I told. And thank you for the attention and question. Maybe I speak very fast, I don't know.
So thank you very much, Suelen. Do you have questions to Suelen, or just please open your camera or microphone or only the chat? So Suelen, I have one. As I remember, this compound is under protection by patent, right? So we cannot.
Yes, it's under analysis from Fiocruz nuclear patents. So they told us to don't put the name, so put a code. So I put a code as EMT here.
So you just have a code, but did you have the opportunity, or did you check for the toxicity in animal models? Because it seems the compound it's clearly has efficacy to kill leukemia cells, but is it cytotoxic to the mice, for example? Because assuming it's an NF-κB inhibitor, maybe it will cause a dysfunction in the immune system or something like that. Did you check for this type of?
In our studies, we did some, but it's not complete, so I don't show here. But we see the weight of the mice from the control and for the treatment, and they are similar. They don't lose a lot of weight, so this can show indirectly the toxicity. And also we made a histological tissue to see in the microscope the tissues, but this is not done yet, so I can confirm, but in the future
so
about the toxicity.
Someone else have a question, please?
I would like to congratulate you, Suelen, for the very nice seminar. Very nice thesis. Good luck in your project.
Yes, it's a very interesting to see novel compounds, because sometimes we do sigma pharmacology, and when we have a novel compound, it's very exciting. So thank you, Celine, and congrats for the presentation and for accepting our invitation. So the last oral communication in this cancer symposium comes from the student, the PhD candidate, Marina Del Gobo. Marina is a student from the Federal University of Santa Catarina, the Department of Pharmacology. Mrs. Del Gobo will present her work on NRF2 in lung cancer and the protein named thyrodoxine reductase. And the presentation has data from patients, and since maybe it's interesting for this audience. So Marina, thanks for accepting the challenge to present for our scientific community. It's your turn. Take your time.
Okay, thank you. Can I start? Right, okay.
Yeah.
So good afternoon, my name is Marina. I'm PhD student in pharmacology at the Federal University of Santa Catarina. And now I'm going to present some of my thesis results, which is NRF2 pathway activation and tumor recurrence correlate with thyrodoxine reductase 1 upregulation in non-small cell lung cancer. So lung cancer is one of the most common malignancies and the leading cause of cancer-related deaths worldwide. In 2018, the global incidence of lung cancer was 11.6%, and global deaths by lung cancer were 18.4%. There are two main types of lung cancer: small cell lung cancer and non-small cell lung cancer. And understanding the difference between each type is critical to treatment decisions and outcomes. Non-small cell lung cancer is the most common type of lung cancer, and approximately 85% of all lung cancers are characterized as non-small cell lung cancer. So, the non-small cell lung cancer category, there are three subtypes, like adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. This tumor type has different mutations. So 30% of the mutations occur in the NRF2 pathway. The risk factors for lung cancer is smoking is the leading for the lung cancer, and it's responsible for 18% of lung cancer deaths. So these smokers have high oxidative stress, as well as constantly higher levels of NRF2. So in this previous work from our group, it was shown in the Kaplan-Meier representation of NRF2 pathway signature of lung adenocarcinoma. And cancer types with higher expression of NRF2 signatures and targets in the high-risk group were assumed as a potential candidates for NRF2 pathway involvement in the disease outcome. And phenotype of resistance to oxalate agents. And when you look at the gene with the high risk of the antioxidant genes in the TCGA cohort, thyrodoxine is the highest gene. So the higher the expression of thyrodoxine reductase 1, lower the survival of patients with lung adenocarcinoma. The thyrodoxine reductase 1 pair is an important modulator of protein redox homeostasis, but also exerts a number of additional redox regulatory functions. In context of cancer, the thyrodoxine reductase 1 pair has emerged as a potential target to cancer therapy especially in tumor types susceptible to oxidative stress. NRF2 is one of the most important regulators of detoxification and oxidative stress responses in mammalian cells, and a master regulator of thyroxine reductase 1 gene expression. In non-stress conditions, low cellular levels of NRF2 are maintained via proteasomal degradation through binding to KIP1. In somatic mutation in these genes occur in approximately 25% of lung squamous cell carcinomas and 20% of lung adenocarcinomas. So these alterations may affect the KIP1 NRF2 protein-protein interaction, leading to NRF2 accumulation and constitutive activation of NRF2-dependent genes transcription. And these characters are a subset of non-small cell lung cancer. So in this research, we selected thyroxine reductase 1 from a screening of 64 oxidative stress-related genes, expression, and survival data of lung cancer patients from 35 published data sets. And we made the bioinformatic analysis into CGA samples. We used a hospital-based cohort of 65 non-small cell lung cancer patients coming from the Hospital São Vicente de Paulo, Passo Fundo, to make the immunohistochemistry to thyroxine reductase 1. And we made an analysis in vitro using lung cancer cell lines, like KIP1 mutant cell lines. So the results are high thyroxine reductase 1 levels predict lung cancer recurrence. So we selected a method to show a good signal to background ratio. Thyroxine reductase 1 signal is compared to negative control without the primary antibody. So the thyroxine reductase 1 signal was also detected in the cytoplasm of thyroxine reductase 1 low samples and in both cytoplasm and nuclear compartments of high cells of thyroxine reductase 1. And thyroxine reductase 1 is constitutively expressed in most of the human tissues, and in fact, all lung tumor samples display immunohistochemistry scores higher or equal 1. So high thyroxine reductase 1 immunohistochemistry scores were observed in 38% of the samples. And positive control staining of A549 cells, KIP1 NF2 multi cell lines. So in the Kaplan-Meier these results, the Kaplan-Meier estimate of survival by time, by months, according to the thyroxine reductase 1 scores, high versus low, show that patients' tumor expressed high thyroxine levels and that underwent lobectomy surgery display shorter time to local regional and extent metastasis recurrence as evaluated by means of DFS. It's the measure of time after treatment during which no sign of cancer is found. And DFS in patients who underwent adjuvant chemotherapy after tumor resection. The patients cisplatin in 33 treated patients. To get insight whether the relationship between thyroxine reductase 1 and NRF2 also occurs at a protein level, we use immunohistochemistry to compare NRF2 levels between samples with the highest versus the lowest thyroxine reductase 1 scores in our non-small cell lung cancer report. So the DFS analysis show that high content of both thyroxine reductase 1 and NRF2 is associated with shorter time to disease recurrence when compared to thyroxine and NRF2 low patients. So we next made the experiments in vitro, in vitro evaluation of survival impact of thyroxine reductase 1 in cells with hyperactivated NRF2 pathway. So we next investigate a potential role of NRF2 as a causal factor of resistance to rhodopsin edutase 1 inhibitor in KIP1 mutant cells. And in KIP1 mutant cells line, A549, rhodopsin edutase 1 inhibitor with auranofin is compensated by NRF2 activation as referred for NRF2 protein accumulation in the immunoblot. Sorry. In addition, siRNA mediated depletion of NRF2 and treatment with ML sensitized A549 cells to auranofin. So the effect of NRF2 knockdown and NRF2 inhibitor upon auranofin cytotoxicity in A549 cells as assessed by MTT after 48 hours and 72 hours treatment respectively. As you know, GSH synthesis, recycle, and rhodopsin are the two major compensation TIOL-dependent antioxidant pathways that maintain protein DTOL disulfide balance, and both are under control of NRF2. And in fact, A549 cells succumb to auranofin combined with GSH synthesis inhibitor BSO when compared to either BSO or auranofin alone, in green and in red. So the presence of GSH precursor and TIOL antioxidant NIC completely prevent auranofin cytotoxicity in blue, as well as abolish the deleterious effect of BSO auranofin combinase in orange. On the other hand, the TIOL-unrelated antioxidant Trolox had no protective effects. Interestingly, KIP1 mutant cell lines A549 treated with auranofin 1 and 2.5 micromolar show no alterations in GSH levels, whereas KIP1 wild-type calo-free cells present a significant GSH depletion after 24 hours treatment with the rhodopsin edutase 1 inhibitor. In A549 cells, ML alone caused 31% decrease in GSH, and this effect was not potentially by auranofin treatment. So these results indicate that KIP1 mutant cells are less susceptible to changes in GSH TIOL homeostasis when rhodopsin edutase 1 function is inhibited. Regarding KIP1 mutant cells' response to platinum compounds, unexpectedly, auranofin did not synergize with cisplatin in both 72 hours cell viability. And long-term clonogenic assays, we didn't see that thing. So in contrast, NRF2 knockdown and GSH depletion with BSO made A549 cells more sensitive to cisplatin. Taken together, these findings indicate that rhodopsin edutase 1 overexpression confers neither susceptible to auranofin nor resistance to cisplatin in KIP1 mutant cells. On the other hand, NRF2 GSH axis seem to be a key component of cell survival, proliferation, and chemoresistance in KIP1 mutants. So the conclusions of this work is amid 64 redox genes, rhodopsin edutase 1 shows association with poor survival in non-small cell lung cancer. High rhodopsin edutase 1 levels predict poor DFS and DMFS in a cohort of non-small cell lung cancer patients. Rhodopsin edutase 1 expression correlates with NRF2 target gene signature in NSCLC. High rhodopsin edutase 1 overlaps with altered NRF2 KIP1 axis, but not other non-small cell lung cancer mutations. NRF2, but not reducing-nuclease 1, is a key for survival platinum resistance of KP1 mutant cells. So the paper was accepted at the end of the last year at Head for Biology and Medicine, and there was a several interesting results there. You can access the paper by opening your cell phone camera and scan the QR code here. And I would like to thank all the funding and collaboration agents. This is my contact email for any questions and suggestions. Thank you.
Okay, thank you, Marina, for your presentation. It was very good. I'm kind of suspicious to talk about your presentation.
You are conflicting. You have conflicting of interest.
Conflict of interest. Yeah. So, does anyone have questions to Marina? Just have room for one question or two.
I have one. I put in the chat.
How to use compounds targeting redox pathways without affecting normal cells?
What do you think, Marina?
Yeah, that's a good question.
Tell me the solution.
It's really complicated. I don't know how to say you.
Maybe by applying nanotechnology.
Yeah, it's a
Why not?
alternative.
I have a formulation that you can put the
if you connect the first talk from Dr. Bishop to this last one, maybe you have to identify molecular vulnerabilities that cancers that are redox incompetent, such as the BRCA1 mutant cancers that cannot upregulate NRF2, for example. So these cancers may be more susceptible if you affect the redox system. When you go to these cancers, Marina is working the KP1 mutant cancers in lung cancer. Maybe these tumors are very well protected, expressing high levels of glutathione enzymes and other, so they might be resistant to a redox approach, such as you have seen with thyrodoxines. So that's one point, maybe. Do you have any other question? It looks not. So we are running very late, so people have poster sections. I want to thank you again very much, Dr. Bishop, Dr. Braganhol, the PhD students and masters Marina and Swellen for giving time to present here. And we'd also thank very much for the audience that joined us in this meeting. So you should be here in Florianopolis. Today is a beautiful day, sunny day. Maybe the next year we'll have this meeting here in the island in person. So I think now we can close this cancer symposium. Thank you all. Bye.
You can invite me again, Alfeo, that I'm going to Florianopolis. Sure. Alex, do you know Florianopolis?
No.
Hi, you have to come. You have to come.
I was expecting to be there, but it was then now I'm stuck here.
Me too. Porto Alegre, there is no beach, no sun today.
It's not a small city, but it's a very calm city, and so you can still don't have violence. So it's a very good place for a meeting. Let's see you next year. Okay, guys. Bye.
Thank you, Alfeo.
Bye.
Thank you, everyone. Bye. Very good. Bye.
Bye.